the hypothesis for superactivity is that upon unfolding, protein gains some enhanced dynamics, which results in protein superactivity. To further support this idea, we use neutron spin echo, which is a technique to detect protein dynamics and domain motion within the protein. We calculate basically the effective diffusion coefficient between the proteins, and we can apply 3 different models in the first to calculate the Q dependence of diffusion coefficient. In the first model, the protein is assumed as a rigid structure with no domain motions. In the second model, the protein is assumed as n rigid domain with completely independent domain motions. And in the third model, the protein is assumed as n rigid domains with constrained domain motion. This is the result of neutron spin echo. If you look at this top related to the pure lysozyme, this solid red line is related to the rigid body calculations for the enzyme. These dotted red lines are translational, calculated translational diffusion coefficient based on dynamic light scattering. And these are in vitro conformations obtained from SANS. So if you look at the pure protein, you can see that the data is agreeing with rigid body calculations and oscillates around the calculated diffusion coefficient. This shows that in nanosecond time scale, the protein adapts a rigid structure as a native conformation. If you look at 5 millimolar trans surfactant and also up to 8 millimolar concentration under UV light, still the data agrees with rigid body calculation, which suggests that the protein does not undergo dynamic motions beyond translational or rotational. But if you look at this at higher concentration under visible light, we can see that the data deviates from rigid body calculations. Therefore, it means that there is some enhanced dynamic motions within the protein, probably domain motion. So we applied 2 other models, these freely jointed and soft linker domain models. And in both, you can see that there is a peak occurring in the region that our data deviates from rigid body calculation. Therefore, these results altogether suggest that upon unfolding, protein undergo enhanced domain motion, which results in a superactivity of the protein. These results are in agreement with SANS-based measurement. As we can see, in the presence of 8 millimolar trans surfactant, protein dramatically unfolds. Therefore, we can say that protein dynamic is also very important for enzyme catalysis. And as a result, we can, instead of looking at locking key mechanism of enzymes, we can now look at dynamic form-function relationship of enzymes in modern enzymology. So to summarize everything, in both cases, carbonic anhydrase and lysozyme, we saw that we can control the structure of the protein reversibly with light illumination. In both cases, the degree of unfolding in the presence of trans surfactant is much more than that of the cis. However, in the case of carbonic anhydrase, protein unfolding results in enzyme inactivation, but in the case of lysozyme, protein unfolding results in superactivity, which arises from enhanced dynamic and domain motion within the protein. Therefore, altogether, these results suggest that we can reversibly control protein structure, protein dynamics, and protein functions using photoresponsive surfactants and light illumination. Some advanced application of this study includes, for example, dissociation of enzyme inhibitor complexes to reactivate inhibited enzymes. It can also be applied to dissociate aggregates to regenerate functional proteins. Before I end my talk, I would like to thank my advisor, Dr. Tedley, for all his supports and helps, and I would like to thank you all for your time and being here today for my presentation.
Questions? So you show that there's a response with the concentration of surfactant. Is there a dose-related response to the dose of UV? Or is there a threshold? We explored that.
This is the effect of UV on the surfactant, basically. Do you mean
so is there a response of the the amount of conversion from trans to cis with different levels of fluency, if you will, what would be?
Yes, actually we have measured the ratio of the amount of cis to trans up in UV light illumination. Basically, under UV light, more than like 90% of our surfactant is converted to cis isomer. Under visible light illumination, the ratio is almost about 75 to 25, trans to cis. And in both cases, as the photoisomerization occurs, that strongly affect our results. In the case of activity, it changes the slope. In the case of conformational changes, it comes with protein refold or unfold, depends on the wavelength of the light we use. Does that answer your question?
Sort of. Depending on the exposure of UV, can you modulate that, the effect? Like,
You mean by
similar to
versus
how
time?
you are with the concentration. Exposure or,
So you mean versus
brightness.
time?
Yeah, brightness. Intensity is the right word.
So you mean
Sort of the ultraviolet, how much, lumens you absorb.
Something like that.
we actually I never tried that, but what I tried is I measured the time required for the complete photoisomerization, and I counted for that. So I assume that when the complete photoisomerization is occurring, my results is based on that point and up. You know what I mean? Does that make it more clear?
Any other questions?
Well, what about the kinetics of the unfolding? I imagine there's a lag from when you do the unfolding to when you would monitor the conversion from unfolded to folded. Did you study that at all?
The kinetics?
Yeah.
For enzyme activity, I studied the kinetic measurements, which is like, do you mean?
So if you unfolded it and then at times you were start making activity measurements for the enzyme, would you see any lag for the protein folding part of it, or if there's changes actually?
Well, basically, it happens very fast. I was saying nanoseconds. Once I shine UV light, it depends on actually the condition that we use, surfactant concentration. Higher the surfactant concentration, longer the time for photoisomerization. But altogether, it happens very fast. I would say, like, if I use just regular UV light illumination at higher surfactant concentration, for complete photoisomerization, it takes about like 10 minutes, maybe less.
Okay. I was interested if you had relatively complicated proteins, some of the ones you work with take hours once going from unfolded to folded state. There would be a significant lag when you would activate or deactivate. Yeah.
Excuse me. So why do you compare the protein CA to lysozyme?
Protein what?
Protein CA and the lysozyme. Why do you compare?
Why do you compare?
Yes.
Because the effect of unfolding is different. In both cases, we saw that we can control the protein unfolding, but in the case of carbonic anhydrase, unfolding results in inactivation. But in the case of lysozyme, it results in superactivity, which is two complete situations. So we were interested why unfolding affects in different ways. So we concluded that this is because of, in the case of lysozyme, is because of protein dynamics, which we concluded that dynamics are also very important for enzymes, enzymatic courses.
All right, well, let's thank Panteha again. And while our next speaker is setting up, maybe we'll take like a 2 to 3 minute break, stretch our legs. The judges want to go get a cup of coffee.